Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Right now, the most critical manufacturing facilities on Earth, they're completely invisible.
Yeah, they really are.
And they operate at a scale that just totally defies imagination. I mean, we rely on these microscopic biological factories, specifically, strains of E. coli, to synthesize basically the entire backbone of our modern bioeconomy.
Right.
We are talking life-saving therapeutics, synthetic insulin, complex bioplastics, even the food-grade enzymes that end up in the groceries you buy every single week.
Exactly. And, the challenge isn't actually getting the bacteria to produce what we want. I mean, we solved the basic genetic insertion part of that decades ago.
Right. The basic science is there.
Yeah, the real friction, the thing holding the industry back, is getting them to execute that production efficiently and safely at a massive global scale.
Right.
The current standard operating procedure, it basically relies on brute forcing the issue, which it creates this whole cascade of physiological and economic failures.
Which brings us directly to the source material for our deep dive today. We're looking at a highly technical dossier, and it's simply titled IPTG-Free Expression Strategies.
A bit of a dry title, I know.
Yeah, but the mission here is anything but dry. We are trying to decode how bioengineers are basically hacking E. coli to make protein production cheaper, infinitely safer, and just, completely autonomous. We're translating this dense bioprocess manual into a sort of masterclass on microscopic engineering. So, okay, let's unpack this, because to understand the brilliant workarounds these scientists have come up with, we kind of have to look at the central antagonist of this whole industrial story.
The villain, essentially.
Exactly. We need to talk about the chemical trigger known as IPTG.
Right. So IPTG is the industry standard, has been for a long time. In classical BL21, DE3 type bacterial hosts, which, by the way, are the absolute workhorses of biomanufacturing, you need a way to turn the factory on.
Right. They don't just start making the drug automatically.
Exactly. You grow the bacteria up to a certain density in this massive vat, and then boom, you drop in the IPTG.
And mechanically, the dossier explains that IPTG mimics lactose, right?
Yes, exactly. It binds to what's called the lac repressor, fundamentally changing its shape so it basically just falls off the DNA.
Oh, wow.
Yeah, and that clears the road for the T7 RNA polymerase to rush in and begin fiercely transcribing your target gene.
But the dossier highlights some pretty severe platform-level concerns with this mechanism. I mean, I keep looking at the data on how the bacteria react, and I can really only describe IPTG as the ultimate toxic middle manager.
That's a perfect analogy.
Right, because you drop this chemical into the vat, and it essentially screams at the bacteria to drop everything else they're doing and work exclusively on this one product.
Yeah.
And because IPTG is a structural mimic of lactose, not actual lactose, the bacteria can't actually metabolize it.
Right, they can't break it down.
So they can't turn off the signal. The alarm bell just keeps ringing indefinitely.
The metabolic drain is absolute. I mean, the T7 RNA polymerase is so powerful and so fast that it completely hijacks the host's ribosomes.
Wow.
Yeah, the bacteria are forced to divert all their energy and amino acids away from their own survival, away from replication. The dossier specifically points out the immediate consequence here, which is severe growth arrest.
Meaning the population literally stops expanding.
Completely. Because they are just utterly exhausted by this synthetic demand.
And when you push workers that hard, naturally, the quality of the product collapses.
Oh, inevitably.
The translation machinery is moving so rapidly that the newly formed proteins, they just don't have time to fold correctly.
Right.
Their hydrophobic regions get exposed, and they start aggregating into these dense, useless clumps inside the cell.
Inclusion bodies.
Exactly, inclusion bodies. You've essentially ruined your own yield.
Right.
Because the chemical trigger forced the system to run way hotter than the physical constraints of the cell could handle. But, what's fascinating here is that the biological stress is really only half the equation. Okay, what's the other half?
You have to evaluate the downstream operational reality. ITCG is a highly complex synthetic molecule. If your bioreactor is churning out an industrial solvent, well, maybe you don't care about trace chemicals.
Sure.
But if you are manufacturing a food enzyme or an injectable therapeutic like a monoclonal antibody fragment, injecting a synthetic inducer into the process introduces a colossal regulatory burden.
Because you have to prove to the FDA that it's gone.
Down to the parts per million level. The regulatory bodies require exhaustive documentation. rigorous impurity profiling to ensure not a single trace of that synthetic chemical makes it into a human body.
That sounds incredibly expensive.
Oh, it is. Running those continuous mass spectrometry and HPLC validations, it adds layers of purification steps. You're extending your downstream processing time, which drastically inflates your operational costs.
Wow.
And that's before you even factor in the upfront cost of purchasing industrial quantities of IPTG itself.
So the industry is basically trapped. You have this chemical trigger that causes growth arrest, guarantees a percentage of your yield will just be lost to inclusion bodies, triggers regulatory audits, and costs a total fortune.
Exactly. It's a bottleneck.
So the dossier outlines several strategies to completely bypass this system, and the first biological workaround relies on, I think, the simplest environmental lever you can pull.
Right.
If the chemical trigger is ruining the workflow, you just alter the nutritional environment of the vat itself.
Yeah, this shifts the paradigm from an active chemical trigger to an environmental cue, utilizing what's called auto-induction media.
Okay.
We're moving away from brute force and leveraging the organism's own evolutionary programming. The strategy takes advantage of a mechanism known as carbon catabolite repression.
Which is fascinating because it's basically like a sequenced buffet.
A sequenced buffet, I like that.
Yeah, because the media described in the source, it doesn't just flood the tank with one food source. It provides a really carefully calculated ratio. You've got glycerol for the baseline energy, a very restrictive tiny amount of glucose, and then a large reserve of lactose.
Exactly. And the bacteria will always prioritize glucose.
Yeah.
Always. It is the most thermodynamically efficient energy source available to them.
Makes sense.
So while glucose is present, the cells produce a regulatory protein that actively locks down the lac operon. They are willfully blind to the lactose in their environment.
Wow.
During this initial phase, the entire population is singularly focused on multiplying. You are rapidly building a massive, healthy workforce without any of the stress of protein production.
When they hit the wall, the trace amount of glucose just runs out.
Right.
They experience that brief pause. I think the dossier calls it the dioxic shift.
Yes, the dioxic shift.
Where they have to assess the environment and basically realize, uh-oh, we need a new food source.
Exactly.
Yeah.
And that depletion of glucose causes a spike in cyclic AMP inside the cell, which totally alters their transcription profile.
Oh.
Suddenly, they unlock the ability to import the lactose. Once inside, the lactose is converted into allolactose, which acts as the natural endogenous trigger for the T7 RNA polymerase. So the vat seamlessly transitions from the growth phase into the production phase.
Without a single human intervention.
Exactly. Zero human input.
The numbers in the dossier really back up how effective this natural transition is, too. For a lot of target proteins, the expression yields hit like 80 to 100% of what you would achieve using the toxic boss, IPTG.
Which is incredible.
Right. And because the induction is gradual, scaling with the population's metabolism rather than a sudden chemical shock, the bacteria don't undergo that violent growth arrest.
Right. The chaperone proteins actually have time to properly fold the target molecules, which drastically reduces those inclusion bodies.
So it fundamentally changes the operational logistics. You just seal the bioreactor, set the environmental parameters, and let the thermodynamics of the bacterial metabolism dictate the timing.
Exactly. It's elegant.
But the source material takes this nutritional hacking a step further. It explores using other less common sugars like L-rhamnose and L-arabinose, which are highly appealing because they act more like a rheostat, like a dimmer switch rather than just a binary on-off.
Right, you can tune the expression rate.
Exactly, to match the folding capacity of your specific target. And here's where it gets really interesting.
Oh, yeah. I know where you're going with this.
The dossier introduces a complex inducer alternative that legitimately sounds like a typo the first time you read it. Skim milk.
Yes. The use of complex, unrefined agricultural streams as an induction method.
I mean, wait, so instead of buying chemicals that cost hundreds of dollars, we are literally taking a highly calibrated, heavily monitored stainless steel bioreactor and giving the bacteria a milk break.
I mean, essentially, yes.
The data actually indicates that adding just 1 to 2% sterile skim milk to the culture can induce reporter expression on par with high-grade IPTG. That's wild.
It is wild. But if we connect this to the macro level, let's analyze the economic logic there. Skim milk is essentially just an unpurified, complex delivery system for lactose and trace minerals.
Right.
It triggers the exact same allolactose conversion pathway we just discussed. In the context of producing food-grade enzymes or cultivated dairy proteins? Utilizing a commodity agricultural product as your inducer is incredibly strategic.
Because it completely sidesteps the regulatory nightmare.
Exactly. The inducer is already a universally recognized, safe food substance. You just bypass the grueling impurity profiling required for synthetic chemicals.
That's brilliant.
You are replacing a reagent that costs exorbitant amounts per gram with an agricultural byproduct that costs pennies by the gallon. It really demonstrates how looking outside standard molecular biology catalogs can solve massive industrial bottlenecks.
But the dossier does acknowledge a massive caveat with these nutritional hacks, right?
Right.
Auto-induction media and complex inducers like skim milk, they're highly dependent on the physical infrastructure of the manufacturing plant.
Yes, that is the catch.
Like, some industrial setups are locked into highly specific proprietary nutrient broths. If you're operating a continuous flow bioreactor, you can't just change the media composition on a whim without throwing off the entire chemical balance.
Right. The external environment is sometimes rigid.
Yeah.
You cannot always dictate the carbon source. And when the environment is locked, the engineering must move inward.
Right.
You have to build the sensory trigger directly into the genetic architecture of the organism.
You put the switch inside the bacteria itself.
Exactly.
The dossier refers to these as self-inducible architectures, and it highlights a highly sophisticated approach called the SILEX system.
Right, SILEX, which stands for self-inducible Lexa-based expression, though the dossier focuses on a distinct stress response pathway to achieve this autonomy.
Okay.
In this model, the host E. coli is equipped with an engineered plasmid that drives the expression of a specific chaperone protein, HSP70.
But the critical design feature here is that the control over the HSP70 gene is intentionally leaky.
Yes, leaky promoters.
Like, the promoter isn't completely watertight. Even when the system is supposedly dormant, the RNA polymerase occasionally slips in and transcribes the gene, creating this very slow, continuous drip of the stress protein.
And that basal transcription rate is actually the timing mechanism. It's genius. In the early stages of the culture, the cell density is low, and that slow drip of HSP70 is easily diluted as the cells rapidly divide.
Right, because they are multiplying so fast.
Exactly.
Huh.
However, as the population enters the midlog phase, as the vat becomes densely packed and the cellular machinery slows down slightly, that leaky expression begins to compound.
Oh, wow.
The intracellular concentration of HSP70 reaches a critical threshold.
I look at that compounding accumulation like an internal alarm clock. The bacteria are essentially measuring their own population density through the buildup of this protein.
Yes.
Once the HSP70 hits that threshold, it triggers a physiological state shift that finally activates the T7 RNA polymerase. You don't monitor the vat. You don't inject a chemical.
Yep.
You don't sequence a sugar buffet. You just let them divide in whatever standard broth you're forced to use, and the alarm clock rings exactly when the factory floor is at maximum capacity.
From a systems engineering perspective, it is a brilliant decoupling. I mean, if we connect this to the bigger picture, the induction becomes entirely medium agnostic.
Right.
A pharmaceutical manufacturer doesn't have to overhaul their entire supply chain of nutrient media just to escape the IPTG dependency. They implement the SILEX strain, and the biological algorithm handles the timing. It mathematically guarantees the induction happens at the optimal biomass density.
It's incredibly elegant. But the dossier pushes further because it recognizes a structural vulnerability with systems like SILEX.
Right, the plasmids.
Yeah, they rely on plasmids, and relying on plasmids at an industrial scale introduces a whole new type of instability. Plasmids are extrachromosomal, they're separate from the bacteria's core DNA.
Right.
They're essentially heavy blueprints the bacteria have to just carry around.
And the metabolic burden of maintaining those high copy number plasmids is significant. If the selective pressure isn't strong enough, the bacteria will simply drop the plasmid to conserve energy.
Which the source calls plasmid curing, right?
Yes, plasmid curing. And within a few generations, your massive bioreactor is suddenly full of barren E. coli, consuming expensive nutrients but producing absolutely zero of your target protein.
To prevent that curing, manufacturers are forced to dump antibiotics into the vat.
Right, the poison pill.
Yeah. The blueprint comes with the poison pill. If the bacteria drop the plasmid, they lose their antibiotic resistance and die. But now we are right back in the same trap we had with IPTG.
Exactly.
Adding industrial quantities of antibiotics introduces massive cost, intense regulatory scrutiny, and complex downstream purification to ensure the antibiotics don't end up in the final product.
It's a vicious cycle, which is why the ultimate evolutionary step for biomanufacturing is the complete elimination of plasmids and external selective pressures. You integrate the production architecture Directly into the chromosomal DNA of the host. You hardwire the factory.
The dossier highlights two approaches to this hardwiring. The first utilizes constitutive hybrid promoters.
Right.
Scientists fuse fragments of the T7 system with incredibly potent regulatory elements, like the CPC-B300 promoter derived from cyanobacteria.
Or the TT20M1 variant, yes.
Right. And this design basically abandons the concept of a switch entirely. There is no trigger. The promoter is permanently locked in the on position, driving continuous, unrelenting expression of the target protein.
And the second, more comprehensive strategy is the chromosome-based T7 system. The engineers physically splice the genetic code for the T7 RNA polymerase, along with the specific target gene, straight into the E. coli genome.
Wow.
It ceases to be an external instruction and becomes a fundamental inheritable trait of the organism. You require 0 plasmids, 0 chemical inducers, and 0 antibiotics.
Let me push back on the biological reality of that, though. If the promoter is permanently glued into the on position, and the bacteria are violently transcribing this target protein from the second they divide, doesn't that inherently exhaust them?
Oh, absolutely.
Isn't this just recreating the exact same metabolic drain and growth arrest that we saw with the toxic boss, IPTG? Like, we've just moved the boss inside their own DNA?
That is a fantastic point. And you're right, the metabolic tax of continuous expression is immense. If you splice a highly active constitutive promoter into a standard wild-type strain of E. coli, the culture will likely collapse under its own weight.
Right.
Or the bacteria will rapidly mutate to silence the gene just to survive.
So how do you keep the factory from shutting itself down?
It demands an entirely different tier of strain engineering. You are no longer just dropping a gene into an off-the-shelf host. You have to actively evolve and select for mutant strains that possess naturally higher tolerances for metabolic burden. You frequently have to permanently co-integrate specific chaperone proteins alongside the target gene, ensuring the host has the permanent infrastructural capacity to fold the continuous stream of product without forming inclusion bodies.
That sounds like a lot of work.
The research and development up front is exponentially higher.
So you're basically building a biologically fortified super worker before you even turn the machine on. And the dossier makes it clear that the major trade-off for this stability is a complete loss of flexibility.
Yes, that is the cost.
With a plasmid system, if your facility needs to switch from making an enzyme to making an antibody, you just flush the vat and drop in a different plasmid. With chromosomal integration, that organism is locked into manufacturing one specific molecule for the rest of its genetic lineage.
But, for massive high-volume production, that lack of flexibility is largely irrelevant.
Really?
Yeah. If a facility is tasked with producing bulk generic insulin or a high-demand industrial solvent, they don't want flexibility. They want absolute, unyielding consistency.
That makes sense.
The genetic stability of a chromosome-based system allows for continuous flow biomanufacturing. You never have to stop the bioreactor to clean and restart a batch. You just can continuously siphon off the product while feeding raw nutrients in. The elimination of batch-to-batch variability is the holy grail of industrial engineering.
So what does this all mean for you, listening right now? We've traced the molecular logistics, the dietary hacks, the internal alarm clocks, and the genomic hardwiring. Why does the invisible architecture of E. coli matter to your daily life?
Right, the real-world impact.
Exactly. The dossier actually answers this in its final section, the techno-economic analysis, or TEA.
The TA is fascinating because it translates all of these molecular workarounds directly into macroeconomic impact.
Let's look at the direct math. The traditional chemical trigger, IPTG, costs tens to hundreds of dollars per gram depending on the required purity. When a pharmaceutical company scales up to a 10,000-liter bioreactor running high cell density cultures, the inducer chemical alone becomes a staggering line item on the budget.
It artificially inflates the baseline cost of everything produced in that vat.
But the TEA models the shift to these IPTG-free platforms. When you leverage auto-induction media, utilize agricultural streams like skim milk, or deploy the self-inducing silex strains, your induction cost drops from hundreds of dollars per gram to literally cents.
Cents. It's an incredible reduction.
The data indicates that eliminating IPTG alone lowers the total cost of goods sold to CAR-GS by roughly 5 to 15% across the board.
And, the direct material savings are really just the surface. The TA factors in the cascading operational efficiencies, too.
Like what?
Well, by moving to autonomous or constitutive systems, you eliminate the labor hours required to continuously monitor growth curves and manually trigger the vats. The biological precision removes the human error of mistimed induction, which drastically reduces the rate of failed, unusable batches.
And by eliminating the synthetic chemicals and the antibiotics used for plasmid maintenance, you streamline your entire downstream processing. The facility spends a fraction of the time and money running the high-performance liquid chromatography required for CGMP documentation. A 15% reduction in manufacturing costs for critical biopharmaceuticals, biodegradable plastics, or food additives eventually dictates the macroeconomic reality.
Exactly.
It alters the price you pay at the pharmacy. It lowers the cost of sustainable materials. The decisions made at the microscopic level dictate the supply chain of the future.
The dossier successfully argues that IPTG-free induction is no longer an experimental curiosity. It is an urgent, strategic necessity for any platform aiming to survive the scaling demands of the modern bioeconomy.
We started by looking at a microscopic workforce paralyzed by a toxic, expensive management system.
Yeah.
And the dossier laid out the blueprint for a total biological overhaul. Scientists optimized the nutritional environment to coax the bacteria into production naturally. They engineered internal genetic alarm clocks that allow the cells to independently measure their own readiness. And ultimately, they rewrote the foundational chromosomal code to build permanent, unyielding factories that require zero external input.
It represents a fundamental maturation in how we interface with biology. We are moving away from commanding organisms with chemical brute force and toward building autonomous biological systems.
Right.
And if we look closely at the implications of the chromosome-based systems and the potential for continuous operation highlighted in the TEA, it raises a profound operational question for our near future.
I always appreciate a broader implication. What is the logical endpoint here?
Well, if we are successfully hardwiring microbes to permanently and autonomously synthesize complex molecules without any human intervention, without synthetic triggers, and with absolute genetic stability, are we approaching an era where biomanufacturing ceases to look like a laboratory and starts looking like municipal infrastructure? Consider a city's water treatment plant. It operates continuously, silently in the background, without requiring active intervention. Will the next decade see localized, autonomous bioreactors seamlessly integrated into our cities, endlessly and silently churning out the baseline medicines, industrial enzymes, and sustainable materials we need to survive as reliably as water flowing from a tap?
A shift from highly volatile laboratory experiments to the silent, invisible utilities that power the modern world. That is a brilliant framework to end on. Thank you for joining us on this deep dive. Keep questioning how the invisible mechanisms around us dictate the massive systems we rely on every day. We will see you on the next one.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Urnala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.